All right, welcome everyone. It is Wednesday, March 24th. Welcome to the next Water, Wetlands, and Watersheds seminar for spring 2021. Today we are lucky enough to have with us Dr. Lauren Koenig. Dr. Koenig is an aquatic ecologist and postdoctoral fellow at the University of Connecticut. She has her BS in aquatic biology from UC Santa Barbara and then did her PhD in earth and environmental sciences at the University of New Hampshire. Dr. Koenig's research focuses on understanding the effects of global environmental change on freshwater ecosystem processes. And she's really interested in applying both empirical and simulation modeling to figure out how water, energy, nutrients, and all of that ecosystem stuff cycle through broad-scale hydrologic networks. So we're going to hear a lot about these connections between ecological processes and hydrologic forcing today, I believe. So we're very glad to have you, Lauren. Welcome, and you can go ahead and share your screen.
Okay, great. Okay, how's that look? Can everyone see the PowerPoint okay? Before I get started? Great. Yep. Well, good morning, everyone. Thank you, David, for that introduction and for the invitation to participate in this great seminar series. I'm really excited to be able to interact with you all today, even if it is over Zoom. And so I'm talking to you not far from the Connecticut River, which is shown in this Landsat image as it passes through the city of Hartford, Connecticut. And besides getting kind of the 30,000-foot view of the landscape, I like this image because the sinuous nature of the river itself kind of invokes the temporal oscillations that I focus on in my research and that I'll be talking about today. So if we zoom in on our view of the Connecticut River and drop a sonde in the water, we can actually start to observe the fine-scale temporal oscillations of water chemistry and freshwater processes. So while this signal could represent any number of environmental variables, what I'm showing you here is the temporal variability in dissolved oxygen in the Connecticut River over 3 years. I'll be talking a lot about oxygen and stream metabolism later in the talk. And so what we can see off the bat is that variability is obvious at multiple temporal scales. So for instance, we can observe a seasonal rhythm in dissolved oxygen, which, like for other chemical and biological variables, arises from a combination of internal processes. So for example, processes that might be responding to climate drivers like temperature and light, as well as their interactions with the surrounding landscape. So streams and rivers are embedded within terrestrial landscapes. For example, water flow determines the dynamic connectivity between streams and their catchments. And as a result of this connectivity, lateral fluxes or delivery of nutrient and energetic subsidies are highly important. And so streams are very open ecosystems. And finally, these landscape interactions or the connections between terrestrial and aquatic systems are modified by landscape position, which is formalized, in the river continuum concept. And so together, internal processes and their connections with the landscape influence the temporal variability or the seasonal rhythm of processes like nutrient cycling and ecosystem energetics that we see in streams and rivers. And so in freshwater ecology, with the advance of more reliable, widespread in situ sensors, we're increasingly able to discern temporal patterns and drivers at the scale of individual rivers. I would say conversely, kind of a separate body of research has made a lot of headway considering the ecological function of whole watersheds or whole river networks, mostly through the approach of watershed budgets. Although these budgeting approaches often take a black box approach and ignore any heterogeneity within river networks themselves. And so I would argue that as we become more interested in the ecological role of streams and rivers at broader scales, including landscape scales relevant for management and for integrating rivers into large-scale biogeochemical budgets and ecosystem models, I'll argue it's important to consider these ensemble dynamics across rivers to reveal the structure of variability and common drivers that are needed to extrapolate our results to new locations or to new spatial scales. And so within this context, my talk today will kind of explore two overarching questions. The first, what drives seasonal variation in stream metabolism? And the second, how do heterogeneity and river network configuration give rise to kind of a cumulative function of freshwater networks? And so I've spent a lot of time asking this first question for individual streams, but today I want to focus on a story about primary productivity across rivers and my attempts to understand the energetic dynamics of whole river networks. So for the remainder of the talk, I'll be focusing on stream metabolism. If we again consider the dynamics from the Connecticut River, where we've already noted the kind of seasonal swings in dissolved oxygen throughout the year. If we zoom in further, we can see diel oscillations in oxygen that are a result of the daily fixation of CO2 by gross primary production, or GPP, and the degradation of organic carbon by ecosystem respiration, which I'll call ER. And primary production and aerobic respiration really dominate metabolic energy flow in streams. And we estimate GPP and ER as the production and consumption of oxygen, respectively, which is why I'm showing these oxygen time series. And so stream metabolism is really an integrative measure of biotic activity and essentially encapsulates the pulse of river ecosystems as they create and respire organic matter on a daily basis. And so I'm interested in stream metabolism because it's really this fundamental property of river ecosystems. Metabolic rates interact with nutrient cycling rates in ways that influence water quality. Stream metabolism has obvious links to freshwater food webs and trophic interactions. And stream metabolism also integrates upstream processes, and anthropogenic modification may shift processing of energy, carbon, and nutrients. And so stream metabolism is really central to understanding how river ecosystems will respond to environmental change. So a lot of the work that I'll show today is part of a broader NSF macrosystems project where we've quantified annual productivity and respiration for hundreds of sites through on-the-ground measurements, as well as crowdsourcing among investigators that have previously made these measurements. And so this is really a community effort to compile the largest database of river metabolism to date. And my research is really motivated by how we use these spatiotemporal measurements to start to scale freshwater ecosystem processes from individual streams to entire hydrologic networks. And so specifically for this talk, I'm going to focus on my efforts in that area as they relate to gross primary production, or GPP. And so I've already shown you one example from the Connecticut River, but as you can see from this figure showing these vastly different oxygen time series from just 4 streams across the United States, we now know that there's enormous variability in metabolic activity both within and across stream reaches. And this variability is seemingly a big challenge, so graphs like this are really exciting, but it's also a bit daunting. Okay, so how do we understand how river ecosystems work? But we do expect that primary productivity is mediated by a hierarchy of controls, including light, nutrients, grazing by herbivores, for example. But among these controls, we think the most important constraints across rivers is light. Okay, so light availability varies substantially among sites as well as over time, well beyond the latitudinal variation in solar radiation that's the primary driver of the light regime in terrestrial ecosystems. And that's because in rivers, light is often mediated by terrestrial canopies and is structured in the downstream direction. So in a small headwater stream, like the one pictured here, light might be limited to kind of this narrow temporal window prior to terrestrial leaf out. Now, as we move downstream into larger rivers, the influence of shading by the terrestrial canopy is often reduced. And so seasonal patterns in light reaching the stream may be more closely aligned with patterns in incoming light. Next, primary productivity is mediated or constrained by the river disturbance regime. So in rivers, disturbance is equivalent to the annual hydrograph or changes in stream flow over time, like I'm showing here. And so the hydrologic regime might be characterized by a single dominant snowmelt event, for example, or by regular frequent storm events that are evenly spaced throughout the year. Or it could be characterized by long periods without storm events, like in this arid land stream in Arizona, where storm events are generally restricted to the winter months or possibly the summer monsoon. And so despite the tremendous variability we observe in metabolic rates and primary productivity, we can now measure, model, and synthesize stream metabolism more readily. And we're beginning to classify and categorize some of this reach scale variation. So here I'm showing a figure from a paper led by Phil Savoy. And Phil used a database of annual time series of GPP for approximately 50 streams and rivers in the United States. And he performed a classification and clustering analysis to derive 4 empirical primary productivity regimes. And so when I say regime, what I'm referring to is this kind of characteristic seasonal pattern that's more or less repeatable from year to year. And so these 4 regimes are common to streams and rivers. And so the biggest difference is whether a stream had substantial canopy shading or not. Streams that fell out in this category tended to be smaller with greater canopy cover, and so they followed this kind of spring peak pattern where GPP is restricted to this relatively narrow temporal window. And even in rivers that receive ample light, productivity might be mediated by hydrologic disturbance, which was kind of the second most important variable that fell out here. And so we know that a suite of drivers influences GPP at the scale of individual stream reaches, but we don't yet know how these factors combine to influence GPP at broader river network scales. And so the specific research question that I'll start with today is, does variation in primary productivity within individual stream reaches culminate in an emergent productivity regime for river networks? And so I'm really motivated by asking, to what extent are there distinctive productivity regimes for river networks? And as a first step in answering this question, we scaled empirical reach scale regimes using simulated river networks that are derived from optimal channel theory. And so these optimal channel networks are useful because they exhibit the same fractal properties and physical metrics that we observe in real river networks. And within this kind of statistical river network, we identified focal subcatchments to analyze emergent productivity regimes across a range of watershed sizes. And we took 3 different approaches to map these empirical regimes at the reach scale throughout the river network. And the first scenario assumed that network position drives GPP. So in this case, we assumed that all small streams followed this spring peak pattern, whereas all large rivers in the network followed this summer peak pattern. Our second scenario was similar in the sense that all small streams were assigned this spring peak pattern, but instead we assumed that all larger rivers were actually relatively unproductive For example, high turbidity loads or something that kind of influence light attenuation through the water column. And our third scenario was kind of a stochastic scenario where the probability of observing any 4 of these empirical patterns changes with river width. So any reach within the network can follow any of these 4 productivity regimes, but small streams are most likely to follow the spring peak regime, and large rivers are most likely to follow this summer peak regime. And so especially scenarios 1 and 2 are not very realistic, but we intentionally kind of chose these 3 scenarios because we expect that these 3 model scenarios represent end members that allow us to explore the envelope of possible patterns at the river network scale. So what do we find? Here I'm showing you our initial predictions of the productivity regime for a whole river network. So on the y-axis is network scale productivity for each day of the year. And there's a couple of components to the river network productivity regime that I want to draw your attention to, including the magnitude or the area under this annual curve, and also the timing. So the shape of this curve that gives us information about the biological expansion and contraction in the river network throughout the year. So in this scenario, in the spring, most productivity is coming from smaller rivers, whereas as we enter into the summer months, network scale dynamics are really being driven by larger rivers that are shown in green. And so we can follow this expansion and contraction throughout the year. And so to compare the network productivity regimes across the 3 model scenarios, I'm going to show the annual network pattern on the left, and on the right is the spatial distribution of GPP on the day of peak network productivity that's indicated by the dashed line here. So under this productive rivers scenario, network scale GPP was highest during the summer, okay, around day 200, when large river reaches were highly productive relative to small streams. In contrast, peak network productivity occurred earlier in the year for both the stochastic and the unproductive rivers scenario. So peak network productivity is advanced about 100 days. And in particular for the stochastic scenario, the spatial distribution of GPP is much more uniform than either the productive or unproductive rivers scenario. So while a substantial proportion of annual network scale GPP is accumulated earlier in the year, springtime productivity in this stochastic scenario reflects the metabolism of both small and larger river reaches. We also considered networks across a range of watershed size. And so here I'm again showing, the network scale productivity for any given day, where each row represents a range in watershed size from small watersheds of about 40 kilometers squared to as considerably larger, 2,600 kilometers squared. And so in small watersheds, river network GPP is limited to the spring just because there's not many large river reaches that contribute kind of more of a summer peak. However, if we assume that large rivers are productive, the distribution of network GPP shifts later in the year as watershed size increases and more large rivers are sampled. And so we can show kind of similar patterns in the stochastic scenario, which is perhaps most realistic of the heterogeneity we observe in real networks. And this grid kind of represents, our envelope of network scale patterns. And what I want to emphasize is that even very simple assumptions about scaling empirical rates of productivity can yield a wide range of network productivity regimes that vary with watershed size and the productivity of large rivers. But I also want to highlight that as we moved into these larger river networks, especially, river network productivity is often more persistent throughout the year compared to individual stream reaches, which we think of as being very dynamic on daily scales and seasonal scales. And so this network scale attenuation of the spatiotemporal variability in GPP among individual stream reaches kind of results in this relatively stable annual pattern that might be important for mobile consumer communities, for example, or for seasonal dynamics in watershed nutrient cycling or nutrient retention. And I think this is interesting because this kind of portfolio effect of heterogeneity, I think, is well recognized for population dynamics, but perhaps less so for ecosystem processes. And so we did observe that larger rivers become more important as we increase in scale. But if we do consider the fraction of cumulative network GPP, which is the y-axis in this plot, against the contributing watershed area for our entire simulated river network, we can see that small streams remain important to river network productivity. So in this watershed, catchments less than 100 kilometers squared are actually contributing 20% of annual productivity. And as we might expect, small streams become even more important if we relax this assumption that large rivers are highly productive. So even though at the reach scale, productivity in small streams may be low or may be limited to relatively narrow temporal extents, at the scale of entire river networks, we can't discount their contribution because they make up a large proportion of watershed benthic surface area in most networks. Okay, so I want to kind of pause to bring this question back. And you know, the analysis I just showed with the empirical regimes that we measured. But if we really want to unlock the black box at the network scale and be able to predict network dynamics, we need to be able to tease apart and understand the hierarchy of controls on productivity. And so for this question, I'm going to use real networks to understand how light and hydrologic disturbance influence river network productivity dynamics. And so this part of the talk will kind of be presented in two parts. First, how does spatiotemporal variation in light shape network productivity regimes? And in this part of the talk, I'm going to focus primarily on patterns of potential GPP. And at river network scales, potential GPP rates, which I'll call GPP max throughout the talk, tell us what the windows of opportunity are. And then we can pay attention to additional constraints like hydrologic disturbance within these windows. And so for this part of the analysis, I considered 5 focal river networks that were distributed throughout the United States. And I explicitly picked these networks to represent kind of different extremes in the stream light environment. And so we considered kind of a watershed, the Oak Creek watershed from Arizona, that's an arid land stream, has some riparian canopy cover, deciduous ash and cottonwoods, for example. We considered the Black Earth Creek watershed in Wisconsin. This watershed primarily drains agricultural land, and these segments have very little canopy cover. And then we considered 3 temperate forested deciduous watersheds, the Lamprey River in New Hampshire, the Farmington River watershed in Connecticut, and the New Hope Creek watershed in North Carolina. So, you know, our intention was to model the spatial structure of light and its effects on productivity across these 5 networks. But because of the large effect terrestrial canopies have on stream light environments, we still lack field observations of stream light across broad spatial extents, which challenges this sort of work. So to represent light throughout river networks, we modeled stream surface light. And in this model, we can input site geographic location, so latitude and longitude, leaf area index, incoming irradiance, stream width, and tree height estimates based on LIDAR to create temporally dynamic estimates of light that's transmitted through the terrestrial canopy and reaches streams. So I'll use the New Hope Creek watershed in North Carolina to kind of walk you through our basic approach for simulating river network productivity. I'll point out that this map is colored by primary land use. So the eastern portion of the watershed drains primarily urban lands and is colored in red. And the western half of the watershed drains primarily forested lands and here is shown in green. And so if we apply this model of stream light, we get the following patterns in photosynthetically active radiation, or PAR, over the course of 1 year. And in this plot, for a small headwater stream in the New Hope Creek watershed, gray circles indicate the incoming light above the canopy, and orange circles are the predicted estimates of stream surface light. So off the bat, we can see that light reaching streams is, first of all, much reduced compared to incoming irradiance, and that the timing is different in these small streams. As we move to a larger river reach within this same network, we see differences in the modeled stream light environment in orange. So there are merits and trade-offs to building kind of a simple versus complex models of river GPP, but as a first approximation, we've used a constant photosynthetic efficiency, and building on Lily Kirk's work from the Odom Center to estimate light use efficiencies and to convert estimates of stream light to potential rates of primary production. And so from that model, this plot is showing our first estimate of potential productivity, or GPP max, for the New Hope Creek watershed. And on this graph, each point represents the cumulative potential production from all reaches in the network on a given day. And so again, we can kind of highlight these components of a river network productivity regime, where the magnitude, the annual magnitude, represents the area under this curve. We can consider the timing. So in this watershed, the main windows of opportunity occur in the spring, when GPP max is at its highest. And we can also consider the spatial distribution. So here I'm showing a map of the watershed again, but this time the flow lines are actually weighted by the values of GPP max on approximately day 80, which is the peak network productivity for the year. And so when we consider the spatial distribution of potential productivity, we see that, the lines are fairly uniform. It seems like there's kind of this spatial uniform contribution of GPP throughout the watershed. And I will point out that this watershed in North Carolina, because of its network structure, as well as the terrestrial context it lies within, it's heavily forested. So we estimate that approximately 90% of the stream length is likely shaded based on estimates of riparian tree height relative to river width. So perhaps it's not surprising that productivity is most important during this spring period before terrestrial leaf out. The other watersheds, however, all converged on a pattern where network scale GPP more or less aligns with the terrestrial growing season. So summertime dynamics became more important. And I have to say, I was really surprised by this result that across watersheds that vary in terrestrial biome and vary drastically in kind of their light regime, that we observe very similar annual patterns in the timing of potential GPP, with the exception that we're picking up the signal from the summer monsoon here in the Oak Creek watershed in Arizona, which results in this summer dip in potential productivity throughout the network. However, despite the similar phenology at the network scale, these watersheds exhibited different patterns in the spatial distribution of GPP. So in this series of plots, I'm showing the coefficient of variation of GPP max across all reaches within the network for every day of the year. Okay, so how spatially heterogeneous is the watershed? And so if we look to an example like the Oak Creek watershed in Arizona, spatial variability in GPP max was relatively low. And what this looks like is that across the network, contributing stream reaches really follow a similar or a synchronous pattern in light and therefore in potential productivity. In contrast, if we consider kind of the other end here, the Lamprey River watershed in New Hampshire, there's a lot more spatial heterogeneity as evidenced by this coefficient of variation, especially during the summer months. And so here, heterogeneity is really driven by differences in the predicted light regime among headwater streams. And even though we think of this watershed as heavily forested, there are actually a lot of interspersed kind of landscape patches with greater or less light availability that contributes to this heterogeneity. And so these two watersheds converge on similar annual productivity dynamics, but for different reasons. In the case of Oak Creek, we're looking at synchronous fluctuations in light and GPP max. In comparison, in this watershed in New Hampshire, asynchronous activity and heterogeneity is really resulting in a similar pattern. So similar to our results from the simulated networks, what we're seeing is that heterogeneity and asynchrony throughout the network can result in a relatively stable annual pattern at network scales. Further, we could use these results to make predictions about the susceptibility of networks to disturbance events, such as hydrologic disturbance. So for example, perhaps network productivity in New Hampshire is less susceptible to disturbance events than watersheds that have more synchronous or relatively homogeneous patterns in GPP max. Okay, so beyond thinking about potential productivity, I'll wrap up the talk today by considering how this next constraint of hydrologic disturbance further modifies annual network scale productivity patterns. And so for this part of the talk, I'm really focusing on one watershed, the New Hope Creek watershed in North Carolina. And so to start to address this question, we're creating a network simulation. We're not necessarily trying to predict reality with this exercise, but rather say, okay, if we think geomorphic settings should be a constraint on GPP, if this holds true, what are the broader consequences for productivity at network scales? And as I already mentioned, this watershed has kind of an important land use dynamic where the eastern half is more heavily urbanized. Perhaps in this picture, you can kind of see some of our urban infrastructure in the background there. And in urban areas, when it rains, roads shunt water into receiving stream networks as quickly as possible. And so we see this play out in the North Carolina watershed, where two streams that receive the same precipitation input, but a stream segment with higher surrounding road density, so here on the bottom, exhibits a storm response much more frequently, okay, on the average of every like 5 to 7 days. And based on these empirical patterns, we chose to model discharge for every stream reach in the network, taking into account precipitation and using upstream road density as kind of a runoff ratio. And then we assigned a critical flow value that scours stream biofilms. And so, for example, that would be this red line. And so briefly, before in GPP max predictions, we were really using estimates of photosynthetic light use efficiency, here alpha, and daily estimates of light to predict GPP. We included this relative biomass term, I'll call it, because this is biomass relative to some carrying capacity. In the light analysis, we assumed that biomass was essentially always at carrying capacity. But we can't make that same assumption when we consider hydrologic variability. So just briefly, we ended up modeling this relative biomass relative to its carrying capacity as a function of kind of the flashiness or the flow that we modeled. Okay, so if the flashiness threshold is surpassed for any given stream reach, the relative biofilm, or the biofilm relative to carrying capacity, is scoured by some amount that's mediated by this lambda parameter. Okay, in contrast, on days without a storm event, relative biomass is allowed to recover according to a specific growth term R and this modified logistic growth equation. And so when we apply this model, we see that road density modifies the response of simulated GPP to storms. So here I'm showing the precipitation regime over the course of 1 month. And in this example, we have, a big precipitation event followed by several smaller events. And if we consider a few of our stream reaches in this watershed, one reach with low local road density does exhibit, initial scouring with this storm, but is allowed to recover over the course of the month. If we move to a watershed with higher road density, we see kind of a similar dynamic of scour and growth and recovery, but then even these smaller events have a big effect on predicted GPP. So from this sort of simulation, we hypothesize that human land use is an important filter that modifies the response of stream biofilms to storms. And this is important in the sense that disturbance really shifts the spatial arrangement of productivity. This is a graph I showed before of GPP max distributed throughout the watershed on the day of maximum network scale productivity in the spring. And once we account for the effects of hydrologic disturbance, what you can see is that the urban parts of the watershed are so flashy, they're essentially very unproductive. And most of the network scale productivity is coming from the forested parts of the watershed. Now, the hydrologic disturbance also, of course, modifies the annual magnitude of network scale productivity. So here I'm showing the accumulation of potential productivity at the network scale for over the course of the year. And the multiple lines here are showing 5 different years that we simulated. And here I'm including the inset of our kind of potential productivity regime as a reminder. So once we include the effects of hydrologic disturbance, what we see is that network scale productivity is reduced by 50 to 60% compared to potential productivity based on light alone. Further, we see a lot more interannual variation. Okay, so interannual variability in precipitation and the timing of storms becomes important. So as one example, 2016 is shown in this light blue line here. 2016 was an average rainfall year that doesn't stand out among the others. But it has kind of relatively low annual productivity. And what makes 2016 stand out is it had relatively more storms that occurred during the springtime, which is really, this window of opportunity in this network. And so, these effects of hydrologic disturbance, we hypothesize have implications for food webs. Okay, primary productivity fuels the base of freshwater food webs in many instances. And so the kind of spatial distribution, magnitude, and timing of productivity can affect community dynamics potentially. And so I don't have time to kind of go into this part of it, but we've run sensitivity analyses using these models to test whether predicted GPP at the network scale is more sensitive to the resistance or the resilience of stream biofilms. In other words, is the model more sensitive to how much biomass is lost due to a flood, or due to the recovery of productivity after a flood. And we found that post-disturbance recovery rates are a really important parameter dictating broad-scale patterns in river GPP. But this represents an important unknown. Okay, so we just don't have much data on the recovery of stream metabolism beyond a handful of rivers. And so this is really, so this model result is really driving empirical work that I'm starting now. And so our initial motivation was to determine to what extent there are distinctive productivity regimes for whole river networks. And what I hope I've shown you today is that the envelope is broad, okay? And so this can encompass a range of seasonal productivity patterns. But the variation in network scale productivity regimes may be greater within individual networks due to interannual variability and the effects of hydrologic disturbance, not to mention other factors like grazing that we haven't considered, than across river networks. And so it's important to tease apart this hierarchy of controls on river metabolism and the consequences of those drivers at broader scales. And so our approach uses simulation models to link fine and broad scales, and as a tool for exploring the hierarchy of controls on stream biogeochemical processes, such as metabolism. And I think this is important because rivers are at this place where it's a really exciting time to study freshwater systems. We can now observe physical, biological, and chemical processes over new spatiotemporal domains of observation frequency from in situ sensors, as well as geographic extent from advances in remote sensing. And so I'll conclude the talk by just emphasizing that we need conceptual and analytical frameworks to integrate these scales and to better connect the many thousands of measurements made in individual streams to emergent spatiotemporal patterns for broad-scale hydrologic networks. And so I just want to thank kind of our funding sources, NSF, as well as the collaborators that contributed to this project. And if there's time, I'd be happy to take any questions. And thank you very much for your attention.
Thanks so much, Lauren. That's an excellent talk. So yes, please Zoom applause or real applause as you wish. We have a couple of questions in the chat. Anastasia, would you like to ask your question, or do you want me to ask on your behalf? You're welcome to unmute and ask. No mic. Okay, so I'll ask the question for Anastasia. She asks, have you considered the part of the river? So, the upstream, the middle, and leading toward the outlet, how productivity changes for each of these hydrologic profiles? How do you choose locations for measurement and modeling? So I think you addressed some of that, but if you could answer that question.
Yeah, good question. So I think your question relates to how productivity might change throughout kind of the longitudinal shift in rivers. And we have considered that in these models kind of implicitly, in the sense of those statistical river networks. We matched those empirical productivity regimes that really did map out mostly onto elements of river size. To ask how those differences that we would observe in different places in the river network influence these kind of aggregate or cumulative dynamics. So absolutely, that's an important kind of consideration in this work. I think an interesting point you raise is how we might use these patterns to perhaps inform where we should monitor river metabolism. And I think that's an excellent question, and one of our biggest challenges. Okay, so how do we use the temporal or spatial variability to kind of extrapolate our understanding to ungauged or unmonitored basins? And I'm not sure I have kind of the answer for that. I think that one step might be to consider these dynamics in light, for example, like I showed for Arizona and New Hampshire, and perhaps certain networks might have different variability dynamics where we would be able to assess how important is it to kind of capture spatial variability, for example. But I think that's a really interesting question. Thanks for asking it.
Great, thanks. And thank you, Anastasia. I have another question from AJ. He's in the room. So AJ, would you like to unmute and ask?
Sure, yeah. Hey, Lauren. Sorry for my mask. I'm in the lab doing some other stuff and multitasking here, but really, really great talk. Probably unsurprisingly, I was most excited about the kind of end components looking at the effects of urbanization and the resistance resilience components and everything. And I'm really interested to hear more about that sensitivity analysis, but I'm wondering, so urbanization, yeah, it leads to flashier hydrographs and lower base flows, that sort of thing, or higher base flows sometimes, depends on your site. But it also often opens up the canopy, so you have more light hitting the stream because you have less like old forest, old growth. And if you have some engineering going on in your cities, you often have more stable substrate. So riprap, concrete, that sort of thing. In Baltimore and down here, we're finding probably our highest rates of GPP and our concrete line streams, even compared to reference sites. So I'm wondering if you considered or are planning on considering how changes in geomorphology and light availability due to urbanization also feed into that resistance resilience story, or if it's all encapsulated in that model.
Yeah, great question, AJ. And I would love the chance to chat with you more about resistance and resilience at some time. Can everyone still see my slide? I'm actually not sure if I'm still sharing.
I unshared you, but you're welcome to share it again. Sorry, yeah, go ahead.
Okay, I might have to, might take me a minute just to address your question, AJ. So with the statistical river networks, we actually did carry out an analysis where we basically modified the productivity regime. We assumed that if a watershed is urbanized, there would be some removal of riparian canopy cover and that more streams would adopt this kind of summer productivity dynamic. And so here we were kind of asking if urbanization changes the patterns in light, what would we expect? And we kind of found that there was, as you increase or clear more riparian trees in the watershed, not surprisingly, you kind of homogenize on this one summer dominated pattern. But interestingly, in kind of the later half of the talk, when I was exploring the network in North Carolina, that's the real pattern of trees and the real pattern of light. And in that sense, we're just kind of asking about the influence of hydrologic disturbance. So I totally agree with you that there's like multiple ways that urbanization can influence productivity through changes in light and through changes in the disturbance regime. With the latter analysis, since we kind of modeled like the real distribution of trees as opposed to kind of making these assumptions about mapping empirical patterns where, that was kind of implicitly included. But yeah, it's really interesting in terms of, I haven't kind of put some numbers on it in terms of how much would this effect be from light versus hydrology. It's a good question.
Yeah, I forgot that you were using Phil's light model to kind of drive that, so that makes sense. But cool, thanks. Super exciting, super great work.
Thanks, AJ. So, Lauren, I have a question. Sort of, overarching. You're trying to model this thing that is like a integrated measure, right? This potential GPP or realized GPP at the watershed scale. And so, of course, if you could just go measure it, someone would just do that. So, I guess my overarching question is, how do you verify or validate or even calibrate your basin scale predictions? I mean, I love the use of real stream networks, fake stream networks, and just like apply them in all their various permutations and see what you get. But how do you go out and say whether your predicted whole river GPP measures are accurate? And obviously, a lot of work and maybe future work for you, but how would you go about doing that?
That's a great question, and one that kind of is at the forefront of our mind in a lot of these analyses. In aquatic systems, we have this additional limitation in the sense that if we want to scale up our measurements or model kind of whole network dynamics, there's really no way to kind of observe or validate at that scale. Unlike perhaps I'm thinking of kind of a terrestrial forest where perhaps you could kind of compare against estimates from remote sensing to see kind of are you in the ballpark and where the differences might lie. So our approach really relies on validating against measurements made at individual locations, with the sense that if we can accurately predict within reason the productivity dynamics in specific areas of the watershed, we're more likely to kind of have confidence in the modeled simulations. But so far, what I've shown here is kind of conceptual or heuristic in the sense of what are the implications of these drivers that we think are fundamentally important at the scale of individual streams, and what that might mean for broader scales and integrating into biogeochemical cycles at regional scales. I have to say I'm hopeful that in aquatic ecosystems, advances in remote sensing will be able to kind of be harmonized with this kind of intermediate scale approach to provide additional validation measures. But so far we kind of rely on synoptic sampling and synoptic validation throughout networks. But excellent question, and one that will be important to come.
Awesome. Well, I expect we'll be seeing more and more work from you on that topic and in general about stream and watershed scale metabolism. So thank you so much for coming to speak with us today. Another round of applause for Dr. Koenig. And let's see, Sherilyn, who's next for next week? So we can give the announcement out here. CFW. Who's quicker on this?
Alice Besterman.
Okay, so next week we have Alice Besterman, another Northeast, someone else from the Northeast, looking at basically how to restore marshlands by building small little hummocks to support sort of marsh growth. So I think she's from the not Rookery Bay, but from
Buzzards Bay Coalition.
That's
Woodwell Climate Research Center.
Yep. So thanks again, Lauren. It's very nice to meet you. Thanks, everyone. We will see you next week. Cheers.
Thank you all.
And Lauren,